Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing TIFR Biology question paper conducted in the year 2021. If you are new to my channel, please do like, share and subscribe. Then next one. Coming to the year 2021. Making a protein for a gene in eukaryotic cell involves. So what are the steps involved in making a protein from a gene in eukaryotic cells? First, transport of mRNA, then its processing, then transcription, then translation. First, transcription of mRNA, then its transport, then processing, then translation. It should be first transcription of RNA. RNA is produced, then it is processed, then it is transported from the nucleus to the cytoplasm, then translation. So the answer here is option D. The 2020 Nobel Prize in Chemistry was awarded to Jennifer Doudna Emmanuel Charpentier for their work on developing CRISPR-Cas system for gene editing. This system is naturally. So CRISPR-Cas system, it is a mechanism of antiviral defense found in bacteria. So the answer here is option B. At which mitotic stage can standard karyotyping be performed using photomicroscopy of cells? So it is during the metaphase stage where you can perform karyotyping. Then which of the following amino acid do you expect to find in higher proportion within histones compared to cytoskeletal proteins? So in histone, the lysine and arginine amino acids are more. So K is for lysine, R is for arginine. So the answer is option A. Then next one. Biologist Lynn Margulis is a proponent of the theory of endosymbiosis, which suggests that mitochondria of modern eukaryotic cells evolved from bacteria living symbiotically within early eukaryotic cells. Which of the following is supporting evidence for this theory? Mitochondrial rRNA is more similar to bacterial rRNA than to eukaryotic rRNA transcribed from nuclear DNA. That is the supporting evidence for this theory. Answer is option B. Then next one. A genetic cross was set between two heterozygous mice with agouti coat color. Agouti is dominant over normal coat color. The F1 phenotypic ratio with regards to coat color, agouti is to wild type, was 2:1. The possible explanation for this could be. So a cross was made between two heterozygous mice with agouti coat color, and agouti is dominant over normal coat color. So capital A, small a, capital A, small a, two heterozygous mice with agouti coat color. These are the gametes that are produced, and you get capital A, capital A, capital A, small a, capital A, small a, small a, small a. And the phenotypic ratio, the F1 phenotypic ratio was found to be 2:1. That is 2 agouti is to 1 wild type. So possible explanation could be this one, that is capital A was found to be lethal. That is agouti condition in homozygous state is found to be lethal. So the answer here is option B, that is homozygous lethality associated with the agouti locus. Then next one, a group of 3 cells, the P6, P, P5P and the anchor cell are clustered together in the C. elegans hypodermis. Normally, the P6P cell divides 3 times and forms the vulva. You find that if the P6P cell is destroyed, the P5P divides and will form the vulva. If the anchor cell are destroyed, neither the P6P cell nor the P5P cell form the vulva. How do you interpret this data? So in normally P6P cell divides 3 times and form the vulva, but if it is destroyed, the P5P cell will form the vulva. Now if anchor cell is destroyed, now there is a third cell, that is anchor cell. Neither P6P nor P5P cell will form the vulva. So anchor cell is needed for vulval formation. The anchor cell inhibits P6P from formation of vulva. That is not correct. Anchor cell induces vulval formation in P6P cell. That is correct. The P6P cell inhibits vulval formation by the anchor cell, and P5P cell induces vulval formation from the P6P cell. They both are not correct. Bacterial antibiotics can interfere with all of the following except protein synthesis, yes. Peptidoglycan synthesis, yes. Reverse transcription, no. Bacterial antibiotics cannot interfere with reverse transcription. Then next one. A series of beakers of water of different temperatures are placed in front of you. You place your finger in each one and select the beaker containing water of the highest temperature. How did your brain know? The brain receives action potential of different intensities that are proportional to temperature. No. Action potentials are only sent when the temperature of the water exceeds a certain threshold. No, before that even the lukewarm water also you get action potentials. The number of action potentials per second increases as temperature increases. That is more correct. Action potentials are received in a different part of the brain when temperatures are higher. No. Meiosis is used to generate gametes. When is meiosis completed in mammalian females? That is, it is during the fertilization that meiosis is completed in mammalian females. Answer is option D. Then next one. DNA was isolated from wild type Gal plus and mutant Gal minus E. coli cells and separated by density gradient centrifugation. DNA from Gal minus strain acquired a lower position. This indicates the mutation is caused by. Now, the mutation has caused the DNA from Gal minus strain to acquire a lower position. That is, it has become more denser. That means there has been some insertion being taken place. So the answer here is option B, that is insertion. Then next one. A bacterial strain has the following features with regard to growth. Will grow on agar supplemented with arginine and tryptophan. So it must be arginine minus, tryptophan minus and leucine minus. It is not capable of growth on media supplemented with arginine and tryptophan. That means leucine has to be supplemented. Only then it will grow. But leucine is not supplemented in the media. So leucine minus. So it will be leucine minus. Then leucine and tryptophan. It is not capable of growing on media supplemented with leucine and tryptophan. That is, arginine is not present in the medium. So arginine has to be supplemented. That means its genotype will be leucine minus, arginine minus. However, it grows on media supplemented with arginine and leucine. So if you are supplying arginine and leucine and tryptophan is not being supplied, then also it will grow on arginine and leucine supplied media. That means it is tryptophan plus. So leucine minus, arginine minus, tryptophan plus will be the genotype of the bacterial strain. Then next one. The pedigree shown is of 3 generations and shows which individuals suffer from genetic disorder that is 100% penetrant and appears after 40 years of age. Individuals that show the disease are either shaded circles or shaded males. Individuals that do not suffer from the disease are unshaded. Although it should be noted that the grandchildren in generation C are still below 40 years of age. Below this is the restriction fragment length polymorphism of each individual DNA. Individuals A in generation A is labeled as capital A, small a and so on. What is the chance that a child from a union between female CH and the male CI from a family with no history of the disease will never suffer from the disease? Now if you see this, There are two individuals, that is male and female. The male is affected but the offspring is not affected. Here the female is affected and the male son is affected, but the father of the child is not affected. So it is the case of autosomal recessive condition. Now if you see B, that is A small b. A small b is over here. A small b, that means you can see lines like this, one over here and two here. That means it is affected condition. Now AC is here. That is also showing affected condition. Now they have asked what is the chance that a child from a union between female CH. Now CH, if you see, it is affected. CH is affected. You can see three lines in RFLP graph. And if you see CI is this. It is not affected by the disease. RFLP pattern is not the same as that of the affected individual in case of CI, but in CH it is same as that of the affected individual. That is AB, AC and CH are the same. So I told you it is an autosomal recessive disease. So the genotype of CH will be small a and the genotype of CI will be capital A small a. So they have asked what is the chance that a child from a union between female CH and male CI from a family with no history of the disease will never suffer from this disease. So it is 50% chance. So the answer here is option C, that is 50%. Then next one. A protein is purified from a cell extract. SDS polyacrylamide gel electrophoresis is performed for analysis of the protein using treatment with dithiotritol and protease. The image of the gel is shown below. What can be inferred from the treatment of protein with the DTT and protease? Note, the red band in two lanes is the protease used. Protease is a protein, that is why they have given red bands over here to show that it is not the protein of our interest, it is the protease. So DTT, it reduces disulfide bridges. So the protein has two polypeptides, because with the DTT you get two bands linked by disulfide bond. Because it reduces disulfide bridges, there are two polypeptides which are linked by disulfide bonds. You get two bands upon treatment with DTT and two protease sites. That is why you get three bands upon treatment with protease. There are two protease sites on the polypeptide. So the answer here is option B.